Thanks for the invitation to speak today. Thank you all for your attention and attendance. My name is Scott Rudge. I'm a CMC consultant with Syner-G Biopharma Group. I'm going to talk about when and where can CMC be a competitive advantage for you. So if we'll go to I have the clicker, right? So I'm going to break it down into phases like almost all of us do. When you're in pre-IND phase, that's the only time that CMC should be on your critical path, right? You have to get material to do the testing, to do the tox studies, to do the human trial. Getting from lab to first in human fast is critical for value creation, and so this is where CMC can really be a competitive advantage if you can be fast and accurate. During phase one, so what I'm talking about here is when the product is in phase one testing. So this is not making material for phase one, you've made the material for phase one. This can kind of be a slow time for CMC activity. You really don't want to invest too much in a product that you don't know yet if it's going to go forward. So if you use this time wisely, it can be very effective, but it's not usually a time of high CMC activity. In phase two, you should really be preparing for your BLA. It's almost never done, but it's very important if you can be kind of commercial ready with your phase three material. And then during phase three, it's all about execution. And then finally in commercial, it's a high period of MSAT, manufacturing, science, and technology activity. So going into those a little bit more deeply, the pre-IND phase is all about speed. You don't have to be perfect prior to your first in-human studies. You have time to fix mistakes. You have time to do process optimization. You have time to do a lot of product characterization. And if you're working with, a CMO with a 6-month or an 8-month timeline to get you your material, and you're taking it to Australia, then you're paying for speed, but you're not using it. So speed is what you need in the pre-IND. The only mistakes you can make are really big ones, like you didn't get the right molecule, or you didn't get the right expression system, or you didn't pay enough attention to safety, in particular for tox. Most other mistakes you can make prior to phase one are fixable in those other development opportunities. So how do you know you've chosen the right molecule? You definitely want high potency in your proof of concept model, in your animal model, and you want the appropriate developability signals. Now, there's a lot of attention on developability right now. Jane et al. Wrote a fantastic paper. It's in PNAS, the reference is there. And they determined there are 4 groups of assays that you could use to determine whether your molecule was developable. And the 4 groups are shown here, and the assays are listed. And then they looked at how many molecules, and these were all antibodies, by the way, and they weren't the originator's antibody. What they took is the sequences and put them into an IgG1. So they were all in IgG1 format, and then expressed them themselves out of CHO, and then ran these assays on them. So if you had one assay below the thresholds that they show here, they consider that a flag. So you can see most of the approved molecules in the fraction of mAbs on the left-hand side of the chart. 64% of them had absolutely no flags at all, but 15% of them had one failing assay. A little bit higher, 17% had two failing groups, and there's even a couple that had 3 failing groups. I think there are 173 or something like that, 170-ish molecules in their screen. So this was really, really interesting. And a lot of people have been using it and have been using these assays to determine developability up front. If you're not doing an IgG, it can be misleading. And if you come back at 11 for the Ebzina panel discussion, I'll talk about some of the times you break these rules, and for very good reasons. But this is a really good guideline. How do you know you've chosen the right expression system? You want an expression system that's compatible with the molecule, obviously. So if your molecule is an antibacterial, you don't want to express it in bacteria. You know, if it's say a cell wall binder, it's tough to express it in CHO. So that's of course the first screen. Is it compatible with the species or the host? The second is, does the expression system give you something that's consistent? So you can take, say, a stable cell pool and use it for some of your early development and have confidence that when you get to the clone will be more or less the same as the pool. And you'll see here from left to right three kinds of integration technologies for expression: random integration, transposase integration, and recombinase integration. And you see, as you go from left to right, you get really low variability as you go all the way to a site-specific recombinase kind of integration. And when you have that kind of consistency, you can do a lot with your cell pool prior to going to clonality, and take that whole cloning part off your timeline, off your critical path. And then, have you paid attention to safety? And the two biggest things that we see are endotoxin and immunogenicity. And endotoxin you can measure. So you should measure endotoxin. You should think about what your highest dose in tox is, not necessarily your highest human dose, and make sure that you're not going to get a bad signal in tox because you've not been careful with endotoxin in your test article. Immunogenicity has come a long way in 10 or 15 years. There's some great in silico screens for immunogenicity where you can predict anyway. You don't know for certain until you do the animal tests, but you can get a good idea of whether you're going to have an immunogenicity problem before you start expensive animal testing. An important thing is, what are the investors expecting? And they are increasingly focusing on the diligence paid in the early phases of development. And I've seen some wild requests, and it's really not feasible to do everything that they're likely to request. But it's important that you know what they might request, what kind of developability things that they might think that you should do, and then have a reason why you didn't do them if you didn't do them. So risk and speed are a trade-off. If they want no risk, then the speed's not going to be there. If they're willing to accept some risk and you've got some good explanations for why you think the risk is reasonable to accept, the investors will normally come along with you. So through phase 1 through phase 3, I'm going to go through these briefly. There's so much written about drug development after getting into first in human that I'm not going to recap all the nuances of process validation, for example. But in phase 1, you should be planning. This is a time really to design your commercial process. If you can have an idea of what your commercial demand's going to be, its cost of goods going to be important, what kind of demand or what kind of scale up you're going to have to do, that's important. You should also, at this point, make a large supply of a well-characterized reference standard and have an idea of how you're going to assign potency, because that's got to carry you through all the phases of development. You can make mistakes by planning for the wrong scale. You can choose some technologies that aren't as scalable because you're trying to go fast or you're trying to be early. You might stay with a suboptimal container closure for too long. So, for instance, if you have to be in a pre-filled syringe, get into that pre-filled syringe early, like switch from your vial to a pre-filled syringe. if you stay with a suboptimal manufacturer for too long, it can get you in trouble. So if you've picked somebody to get you into phase 1 in a hurry, but you don't think that they're commercial, like they're going to be a commercially acceptable CDMO, then this is the time to find that commercially available, acceptable CDMO and make that tech transfer. And then, you can. I never throw away a sample. I mean, I've never had a situation where a sample stuck in the freezer didn't come in handy somewhere down the line. So if you don't archive your historical samples, and then you have a reference standard change, or you change your cell-based assay or something like that, you're going to need those historical reference samples to continue your project. Potency requalification is an interesting topic, and I could spend another 20 minutes on it. Just want to say that when you change from one reference standard to another, the FDA expectations are really high for how closely that new reference standard has to match the old reference standard in potency. And if you're using a cell-based bioassay, what they want is for the new to call the new reference standard 100% potency of the old reference standard. They want the confidence interval around the new reference standard to include the center point of the old reference standard, and for the entire confidence interval to be within 90 to 110%. Now, most of us have specs that are 70 to 130% just to pass the potency assay. So getting it to 90 to 110 requires a lot of replicates, and we did this for a client recently, where we used statistical package to calculate what the confidence interval range was, and then we compared that to data, and it was actually the statistics were surprisingly close to the real life. So you need to know what your sigma is and put it into that power calculation to determine the right number of samples to test. Otherwise, you risk getting the confidence interval so narrow that you'll miss getting the center point of the old reference standard, or so wide that you'll fall outside of the 90 to 110. So doing the statistical calculation is really pretty important. In phase 2, while phase 2 is going on, you really should be preparing. You should be validating all your analytical methods. You should, if possible, use your phase 3 material campaign to make to initiate your registration stability. If you can do that, then you can take the PPQs that you're going to make later in the development process off your critical path. And you'll be able to file with 6 or 12 or more months of data on for stability. So if you can get your commercial process locked in prior to phase 2, and then execute during phase 2 to make phase 3 batches and use them for your registration stability, you're in good shape. But that means you got to have your specs in place. You've got to have your methods validated. You've got to be ready to go. You should take any major risk that you can think of off the table during phase 2. So anything that you've only done, say, 2 or 3 times, you should test that and make sure that it's good. Get it off the table as a risk for your process, and then replace your ELISA with your cell-based assay, and then stop doing the ELISA. I've got a lot of clients who end up stuck with their ELISA even though they've validated a cell-based method, and it screws them up. If you leave your registration stability for PPQ batches, then your stability, then you're not going to be able to use your PPQ batches for commercial supply, because the stability coming out of those batches is going to overrun your ability to distribute them. So you really need to have your stability started before you do, or ideally, and then you can do your PPQ batches, do your very last thing in filing, and make them available for commercial. And nobody wants to do that, or the common pushback on that is they think the process and the specifications are going to change during PPQ, and so they don't want to start stability so early. I think that's a mistake. I mean, I see very little process change or specification changes after you've started to make phase 3 material. Like, just assume those are going to be your specs, and get enough data to make them your specs. That's my best advice. And then during PPQ or during phase 3, it's all about execution, right? There's nothing more interesting going on as far as I'm concerned anyway. It's about doing the transport validation, doing extractable leachables, doing all the risk assessments you can think of, getting your commercial supply agreements, getting inspections ready. Like, you're done doing science, you're done doing process optimization, you just need to execute on all this stuff. And the biggest mistake you can make in phase 3 is letting the CMC get back on the critical path, because what you want is your filing goes in as soon as the clinical trial's done. You're not waiting for CMC to get stability done or something like that. So during commercial, you want to stay the course. You have to start continuous process verification. You transition to a commercial organization. The mistakes you can make, I think, are widening specifications. I think you should always be trying to kind of like the Six Sigma stuff, you should always be trying to narrow in on more consistent manufacturing. And then assuming the technical support won't be needed anymore during commercialization is a classic mistake, but all too often made. So I think that's it for my talk. Yeah, thank you. Happy to answer a question or two, and thanks again for attending.
Lovely, thank you, Scott. Does anyone have any questions?
All right, thank you.
Thank you.
Yeah.